Welcome to Biology Made Easy. In this lesson, we are looking at dihybrid inheritance or cross. Dihybrid cross involves two individuals and two genes are considered. The two genes each have a pair of contrasting characteristics. Let's say we are having a cross between two genes, the gene for height. And the gene for height, we have tall plants and dwarf plants. That is one gene. Then we have another gene for flower color. In the flower color, we have red flowers and white flowers. So in this dihybrid cross, we have two genes, each one with a pair of contrasting characteristics. And the two genes are on different chromosomes. So the genes are not linked. So you have gene for height here. We have big T and small t alleles. You have gene for flower color here. We have big R and small r on different chromosomes. Now we are considering that tall is dominant over dwarf. So tall is big T, dwarf is small t, so recessive. And red, big R, is dominant over white, small r. So we start the lesson with pure breed tall garden pea plants with red flowers, crossing dwarf plants with white flowers. So parental phenotype, you have tall with red flowers, crossing dwarf with white flowers. And the parental genotype, we have this is tall, red, dwarf, white. They are in different chromosomes. Meiosis occurs in the anther of the male and the ovule of the female, and gametes are formed. This and this can go into one gamete, and no gametes are haploid. This and this can go into one gamete. And 4 gametes can be formed from this. But because it's the same big R, we have only 2 of them. Then this producing gametes, this small t can go with a small r into one gamete, this and this into one gamete. Here we could have had 4 and we have 2 because they are the same. So random fertilization takes place. This gamete can fuse with this gamete and you have big T, small t, big R, small r. This same gamete can fuse with that gamete to have big T, big R, small t. So this gamete can also fuse with this gamete and you have big T, small t, big R, small r. That is this one. So you have these individuals for F1 genotypes. And you can see that because big T is dominant over small t, this will produce tall plants. Big R is dominant over small r, so this will be red. So all the individuals will be tall red phenotype. Now, let's do a dihybrid cross where we cross one heterozygote with a double recessive homozygote. So we have parent phenotype tall red crossed with dwarf white. Parent genotype, we have this and that, tall red, dwarf white. These are the gametes that will be produced. This and this will go to one gamete, this and that, one gamete, this, that, one gamete, this, that, one gamete. Dwarf white, all gametes will be small t, small r. Random fertilization will produce this and that one will produce this and that will produce this genotype, this, that produces this, that produces this. The phenotypes, this is tall red, tall white, dwarf red, dwarf white. Phenotypic ratio is 1:1:1:1. Now, this phenotypic ratio is typical of dihybrid cross if the genes are on different chromosomes. That is, if the genes are not linked. Meaning, these are the chromosomes. This is the gene tall. This is the gene red. These are the genes. If the genes are on different chromosomes, then a heterozygote cross with a homozygote should give us this phenotypic ratio. This is very important to note because if the genes were linked, then we would have had tall linked with red or dwarf linked with white or any other. If the genes are linked, the phenotypic ratio of 1:1:1 will not be obtained. So take note. Now we are continuing. We will look at the dihybrid cross crossing two heterozygotes. And the phenotypic ratios we expect is 9:3:1. So let us go now. So selfing the F1, this is the F1 parental phenotypes, all of them is real. The F1 parental genotypes, this is them. Big T, small t, big R, small r. So that is the F1 parental genotype. Meiosis will produce this gamete. This will go with this one into one gamete. This and this into one gamete. This and this into one gamete. This and this into one gamete. The same thing is happening here. Now, to be able to cross these gametes, the Punnett square is used to show all the possible combinations. This one and this one and this one. And to get the F2 genotypes and phenotypes. So the Punnett square, you start by putting this down. And the gametes, we are going to write them out. The first thing doing a Punnett square is to create this. Put the male gametes on this line and the female gametes on this line. So this is the male gametes here. We have big E, big R. We have big T, small r. We have small t, big R. We have small t, small r. The female also gametes, we have big T, big R. We have big T, big R. We have small t, big R. We have small t, small r. Now we're feeling what happens with this gamete and this gamete fusing. You have this offspring. This gamete and this one fusing. You have big T, big R, small r. This one, you have big T, small t, big R, big R. Here and here, fusing, you have big T, small t, big R, small r. And that's how we fill the whole thing up. I filled in all the rest of the genotype. Now the phenotype. This one is tall red. Tall is dominant. Red is also dominant. So this one is tall red. Here is tall red, 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 tall red. So counting, I have 9 tall reds. And then here is tall white. Tall, this is white. This is tall. This is white. This is tall. This is white. So we have 3 tall whites. 3 tall whites. Then we have here, you see, small TT is dwarf. So this is dwarf red. Small TT dwarf, dwarf red. Small TT dwarf red. So we have dwarf red is also 3. And then we have here dwarf white. So the dihybrid ratio is 9:3:1. 9:3:1. And if you look, we have 2 new combinations of characters. We started with tall red plants and dwarf white plants. So we have tall red and dwarf white. But these tall white and dwarf red are 2 new combinations of character that we didn't start with. And these 2 new combinations are called allelomorphic character. This is a character determined by different alleles. So in the dihybrid cross, we've seen that the phenotypic ratio is 9:3:1 if 2 heterozygotes are crossed. Now, if the genes were linked and were not on different chromosomes, we wouldn't have had this ratio. So anytime dihybrid crosses are done and the ratio 9:3:1 is not gotten from 2 heterozygotes, then there's linkage. So we'll end the lesson here and we'll look at the probabilities of the dihybrid cross in our next lesson. Thank you very much. Goodbye.